Thank you for the introduction, and thank you everyone for being here. I know we're getting close to the end here. So today I'd like to share with you an ongoing study on using machine learning to explore controls of soil carbon globally and within biomes and reveal divergence between observations and process-based models. I'd like to quickly thank my collaborators listed on the right, Avni Malhotra, Will Weeder, Ben Sulman, and Rob Jackson, and also the LTR network for facilitating a working group that motivated this study. Oops. All right, so our understanding of soil carbon processes and our ability to model them have evolved substantially over the last decade. We now recognize that biological, chemical, and physical controls are all important in determining soil organic carbon stocks and cycling. However, only recently have many of these processes been incorporated into soil carbon models at ecosystem to global scales. Furthermore, we know that the relevant controls can vary across spatial scales and biomes, and this can have implications on model development efforts and also on how we use data to constrain models. For example, the controls that may be important for explaining soil carbon variability at the peat-on to landscape scale may differ from those important at explaining soil carbon variability at the landscape to global scale, as in gridded products and model outputs. So in this particular study, I'll focus on a few key climatic and edaphic controls, mean annual temperature and precipitation, vegetation type or biome, and silt and clay content, which will also be called soil texture during the talk. Okay, so over the last decade, we've seen an ever-growing number of soil carbon models. And these can vary widely in their structure and consequently in their response to perturbations, which was summarized very nicely in a publication by Ben Sulman in 2018. And so here I show just a few of these models and how the hypotheses underlying their structure diverge from first order to microbially mediated kinetics and implicit to explicit mineral protection, among other processes. So as we continue to have more and more of these soil carbon models and divergent hypotheses, the question on how to compare, validate, and improve them is an increasingly important one. So in this particular study, I compare three models. The first order, CASA CMP, where the decomposition of the soil carbon pool is proportional to the size of that pool alone. And the microbial explicit, mimics, and corpse models, where the decomposition of soil organic carbon is also catalyzed by the size of the microbial biomass or enzyme pool. And I'll be using model output from the biogeochemical testbed in Weeder et al. Global Change Biology in 2018, where all three of these models were forced with the same inputs. Okay, so first let's take a step back and zoom out into a bigger picture view of soil carbon models and reality. So we have plant inputs or net primary productivity, and this can enter the soil carbon pool. And these inputs can be chemically heterogeneous, varying in C to N ratio, for example. And this soil organic carbon can be decomposed, and a fraction of it can be assimilated into microbial biomass. And of course, these reactions and microbial activity can depend on the mean annual temperature, among other variables. And furthermore, a fraction of this can also absorb to mineral surfaces and enter this passive soil organic carbon pool or mineral associated pool. And of course, the amount that enters this mineral associated pool can depend on the minerals, the clay and silt minerals in the soil. Here we'll denote that, as I said earlier, texture. So of course, in reality and in models, there are a suite of reactions that occur in this box. Let's just take, for example, the mimics model. And this slide is busy on purpose. As you can see, there are many coupled nonlinear equations here that encode this model. But what's important to note is that these environmental controls appear in many places. For example, the net primary productivity enters the soil carbon pools, but can also affect the microbial turnover to capture an implicit density dependence of the microbial community. Mean annual temperature, of course, can affect many reaction rates. And the soil texture or clay and silt content can affect the amount that enters the passive mineral-associated pool, but can also affect other microbial rates. And so there are complex underlying controls within each of these systems of equations. And it's difficult to pull apart these controls. And so we asked here, what is the emergent role of these controls in the models and in the observations? So our goal here was to interrogate observations and physical model output via data-driven approaches to learn the most important driving variables for soil organic carbon globally and across biomes. So specifically here, we focused on 3 observational and observationally derived data products: the soil profile measurements from the ISRIC WOSIS, World Soil Information Service, the Harmonized World Soil Database, which I'll abbreviate HWSD throughout the rest of the talk. And because we expect that the soil carbon is biased low at high latitudes in this database, we also use an adjusted product that uses the Northern Circumpolar Soil Carbon Database where values are available at high latitudes. We then compare these to 3 process-based models, or physical models. The first, as I mentioned, CASA CMP, which is a first-order model, and microbial explicit mimics and corpse models. And here I show current soil organic carbon stocks globally. And while you can see that there is agreement in select regions, it's difficult to know whether the models predict the correct soil organic carbon stocks for the right reasons. So our goal here was to learn the role of these key explanatory variables: net primary productivity, mean annual temperature, and texture, individually on the soil organic carbon from the observations and from the physical model or process-based model output. And we do this via a data-driven learning approach. Of course, we can use a number of different methods here, and we explored a few, but random forest performed best. And so in this particular talk, I'll focus on our results from that. So again, our approach was to use a random forest to learn the soil organic carbon stocks from these key covariates for each data set and model output. And our overarching question was, how do soil organic carbon controls differ globally and between biomes in observations and physical model output? And furthermore, are the global data products representative of the soil profiles? Okay, so first let's take a look globally at these soil organic carbon controls. But before I show any results, I want to quickly define the normalized variable importance that I'll use throughout the rest of the talk. Let's take, for example, 2 variables, J and K. Here, the variable importance of variable K is the mean squared error of the random forest when variable K is removed, minus the mean squared error with all variables, normalized over the difference when each of the variables, here K or J, is removed. We can then repeat this for variable J. And here, these variable importances now sum to 1 across all variables. And the ranking is linear, so it's preserved in the normalization. And this allows an easier comparison between models and observations on the same axis. So in this example, K has more explanatory power and J has less explanatory power. Okay, so let's take a look at our variables of interest. So first, for the soil profiles, what you can note is that there is a large uncertainty here, and this is using 10 random forests with bootstrap sampling. And the average variance explained in the soil profiles was only 25%, which suggest that there are missing controls at this plot scale. But from the variables included, we see texture or soil claim silt content is the most important. Interestingly, in the harmonized world soil database and northern circumpolar adjusted product, we also see texture emerging as the most important. And we note that these vary markedly in their scale and inputs from the soil profiles. And further, we see an increased variance explained here between 50 and 55% in these global data products. So across all the observations, we see texture emerge as the most important variable with the most explanatory power. And in the models, in contrast, we see that mean annual temperature and net primary productivity emerge as the variables with the most explanatory power. I'll also note that the variance explained in the models is much higher, from 50 to 80%. So even a physics agnostic random forest model can capture 80% of the variability in soil organic carbon in the mimics model, for example. And so here we see this mismatch of variable importance between the observations and the models at the global scale. We can take a closer look at the partial dependence plot, at the emergent relationships in models and observations for each of these variables. Here first I show soil organic carbon as a function of clay and silt content or texture. Here we call this conditional here, but it's predicted SOC as a function of texture with all other variables held constant at their mean. And here we can see again, as suggested with the variable importance, that we have a significant and positive relationship here among the observations with texture, but with the models we see no relationship or even negative with texture. So again, the texture dependence was highest in the observations. Here with the same plot with mean annual temperature, again controlling for all other variables, we see here corpse is an outlier. It has the most sensitivity to temperature. And we see that the soil profiles actually have and northern circumpolar database have a higher temperature dependence than the harmonized world soil database, as we expected, because it was biased low at high latitudes. And with net primary productivity, we also see a greater dependence in the models and in the soil profiles here. However, as you remember, the uncertainty in the soil profiles was quite large, and so this result would take further exploration. So again, mean annual temperature and net primary productivity dependence was higher in the models than soil profiles, but not in the gridded observations, which we use often to benchmark these models. So just as a quick sanity check, we can compare these results to a multivariate linear regression, where again, we see that texture dependence is highest and positive in the observations, and that mean annual temperature dependence is the highest in the models. So this confirms our sort of qualitative random forest results, but of course, less variance is explained here than the random forest models, only 10 to 60% in the observations and models in process-based models respectively. And of course, linear relationships are imposed, which we know not to be the case. Okay, so next let's take a look at how these controls differ between biomes and observations and physical model output. So here we first show the results for broad classifications, where we use the Modus land classification and group all forests together in this forest category, and we group grasslands and savannas as herbaceous. What we can see is, again, in the observations, texture has the most explanatory power. And in the models, we see a switch here between the importance of mean annual temperature in forests and net primer productivity in herbaceous biomes. So diving into these sort of broad classifications in greater detail, we'll note that in certain of the forest biomes, we could not explain as much variability. The random forest did not perform well, but in others we did, and in the broad classifications, we had comparable results to the global. And so here I just show 3 of the forest subclasses that performed comparable to the global: the evergreen needleleaf, deciduous broadleaf, and mixed forest. And what's interesting to note is while we see this mismatch globally and in sort of these broad classifications, we actually find agreement in select biomes. Here, deciduous broadleaf, the temperate deciduous broadleaf forests. We can also repeat this for the subclasses within the herbaceous. And again, interestingly, we find model and observation agreement in select biomes, namely temperate grasslands. So to summarize, we show here a potential for novel AI-based benchmarking of physical models and the ability to explore the underlying controls and learn the underlying controls of these models and observations. We see a mismatch of variable importance and emergent relationships between the observations and physical model globally, but we note agreement in select biomes. Furthermore, across the globe and most biome classifications, we see the observations in general motivate a closer look at the representation of mineral protection in models, which is something that has been lacking compared to, for example, microbial microbial decomposition. Furthermore, we see that the predictability of soil carbon variability in soil profiles is far worse than that of the gridded observations and physical model output, suggesting, of course, there are missing plot level climatic and adaptive controls. The reason we could not include these all in this particular study is that these do not appear currently in models, and so additional exploration is needed to identify these and discuss how we would incorporate them in models. And the predictability also varied between biomes, suggesting missing controls or data limitations. And finally, this approach can also elucidate biomes with the largest uncertainty for targeted biome-specific improvements. So with that, I thank you all for your attention. I'm happy to take any questions. Again, I'd like to thank the Soil Organic Matter Working Group I mentioned. I encourage you all to take a look at our website, which has been a huge data synthesis effort among networks in the US and globally. And also thank my funding sources, USDA NIFA and Lawrence Fellows Program at Livermore. So thank you.
Thank you, Katerina. There are at least 2 questions in the chat. So first one, there is no humidity in your analysis. Why?
Right. So as I sort of mentioned at the beginning, mean annual precipitation was something we did initially include. It actually did not change the performance of the random forest models very much, between 1% and 3% of the variance explained. And also it was not included. So moisture controls are not included in the mimics model. And so for this first pass, we only focused on controls that were in all of the models and that showed significant improvement in the random forest performance.
And the second question is, how do you deal with correlated input? For example, M80 NPP might be correlated, so you might have spurious correlations.
Yeah, that's a good question. So that was less. So first of all, the random forest can handle that to some degree. And this is something we have discussed and we are looking into. it did not seem to be much of a problem with the NPP and, excuse me, and mean annual temperature. However, it would be a problem with the net primal productivity and mean annual precipitation, which are quite tightly correlated. So this is something that we are looking into.